Hello, my name is Darwin Chang, and today I will be showing you how to create panel E, or the phylogenetic tree, for the sample summary PDF. I will be doing this with patient 13, MIS-01, and MIS-11. So first you want to watch the previous video about the variance strip chart and watch the first couple of minutes just to make sure that you have installed all the R packages and that you know how to use terminal. This script is very similar to the variance strip chart script in the sense of how to run it. So first we go to the master mutation list scripts under sequencing data and normal mutation burden, and we take the uv also in pychart.r script. We then drag that onto terminal as our first input, and as our second input, we then take the Excel file that we are looking to use to create the pie chart. We simply run that, and we will have a couple of pie charts that are created, two in fact. Let's just open these up. So the first one, which is labeled P13MIS01 truncal, is the truncal branch, aka the mutations that were shared between two single cells. The second one, which is labeled P13MIS01 branchial, is the mutations that are independent of the related cell. In this case, the related cell was P13MAS11, so we will also run the same script, just changing the input a little bit. Just to note, both truncal PDFs should be exactly the same as they are related to each other. You can very easily double check to make sure that they are the same by just quickly looking at the truncal pie charts and just looking to make sure that the percentages are the same. And you can also look at the also in output that was reported. So for P13MIS01, there are 28 also in reports for validated mutations and 28 for MIS11. So we can pretty confidently conclude that these 28 mutations are exactly the same between the two. Sometimes the numbers aren't always shared, so you need to make sure that if they aren't, you double check the master mutation lists for the also in and validation calls, just to make sure that you have the exact same number of truncal mutations. So after that's done, we then take, we don't need this one anymore. We take the one truncal, shrink it down a little bit. Actually, we don't need to do that. We then take MS13 branchial. Make sure you remember which one is which, and then the other branchial. So as you can see, there's already a previous pie chart, but I'm recreating this example. So first, we need to move these texts. There's a small, I guess, line here that you want to make sure you delete by using the direct selection tool and pressing delete twice after selecting the small line. Okay, then we need to make sure that we are in Arial font. So first we That looks pretty good. We can remove the UV and non-UV call. That's only for our purposes in terms of titling. We will have a legend somewhere here to indicate whether or not the mutation was UV or not. So then what you can do is you can take an old phylogenetic tree, grab one of these lines, check the number of mutations, right click, transform, scale, select non-uniform, vertical, and calculate backwards the number of mutations from 31, in this case, to 1. So if we divide 1 by 31, we get 0.0322, so it'll be 3.23%. And so that'll get you the length that ideally you want for one mutation. Obviously, depending on the tree, it could be a varying size because sometimes there are more than just like 100 mutations or something. If obviously you get 1,000 mutations, you have to scale a little bit differently. And if you only have a few, like if you're using UCSF 500 only, then you will need to have probably a larger scale. But it's a good basis to start off of. So we then want to go back to this terminal output. Really quickly, just make sure that we used MAS01 as our first input, MAS13. You then check the number of also-ins and not also-ins to determine the length. So for MAS11, which if you recall was on the left side, we have 115 not also-in mutations. So let's scale to 115 times 100, 4%. And for MS11, on the right, 106. So and then for the middle, or the truncal part, it was 28. So 2800. Just make sure that these sizes are okay. These look good. Kind of just, for now, we're just going to put these lengths next to each other approximately. Grab this width. Just make sure they kind of sort of line up. Width doesn't matter as much, obviously, as the height. It's just to differentiate between the two trees. So first we'll grab this, get a little bit close, then we'll grab the MS11 one. I usually like to separate it vertically a little bit because it creates a little bit less of a mess, personally. We then grab this arrow just to make sure we know, especially if it's not next to the, I guess the line, one of the lines of the tree. So then we need to take the number of mutations. Let's just take this actually. So for the truncal portion, we had 28 mutations. So 28 mutations. Try and center it as best as possible. On the left, we had, I think, 115. Yeah. Okay. Then for the right side, I believe it's 106. Okay, and then you also copy over this legend so that viewers can quickly understand what is going on here, and then take the titles as well. So, if you recall, patient 13, mass 0, 1, and then 11. Okay, that should be it. Let's just really quickly actually carry this over a little bit. Oh, right. Since the truncal is usually, or sometimes is a little bit short, just gotta have the number of mutations a little bit closer. And then we'll drop the legend down here. Copy and paste the whole thing. Let's go back to the actual PDF. Try and separate it from panel D. And obviously center it again as much as possible. Just make sure it's aesthetically pleasing. And that looks overall pretty good. So yeah, this is basically how to do it. Hopefully this has been helpful, and thanks for watching.